Audio recording. Business as usual. To claim the points for the video, you have to write down every time I say an attendance word or phrase. Also take a screenshot for each one. The last word for attendance, if you want to call that, is going to be you asking me a question about the material presented in the lecture. Obviously, you won't need a screenshot for the last one. So by the end of the video, you should have a list of words with a number of screenshots to go with it and a question about the material covered in this lecture. And then you'll email that to me. That's all you need to do for the video. And it is almost 7:50, so you can skip ahead about 10 minutes, and that's when the actual lecture should start. Any plans for the weekend?
Go to the gym more.
Nice.
Example open. Or about to be open.
I have to check. I don't remember exactly when it opens. I'm just going to go ahead and start reminding. Well, I've been reminding everybody of the date because a lot of people didn't take exam too. No, I said it every morning. This is what I do. This is what I do. This is what I do. So I'm hoping.
Pretty good. That's one. Said it's due. That's for 10 times. That's what I do. I got the down before it's this.
Morning.
Morning.
Anybody slaughter any, weekend plans for you?
Besides working now,
Where do you work?
Alta.
Oh, yeah, I think I may have asked you that. Either that or I've had a student here before that's worked at Ulta. It's like a makeup place, right?
Yeah.
I have 3 daughters. So familiar with these things. Things like that and the newest Taylor Swift albums. So I'm very well aware of these things. Morning. 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 That's 3 minutes left. I'll go ahead and do attendance. Start attendance anyway. Rumfeld is not here. Champion is not here. Gross is online. Gross, don't forget, even though I know you're online, you still need to send in all the words, including the one question at the end by 9 a.m. Actually, that goes for everybody, but just want to make sure Gross knew that since he's online. Gunther is not here. Randall is here. Zion is not here. Jackson is not here. Keys is here. Perez is not here. Sigmund is here. Slaughter is here. Smith is not here. Stover is here. Tompkins is not here. Vanessa is not here. Watson is not here. Young, not here. Zweiback is here. Lawrence, not here. Wilson is not here. Have I missed anybody? We still have 2 minutes. Hey, Smith is here. Smith is accounted for. All right, well, since there's less than 2 minutes left, I'll just do the usual repetitive stuff. Next week's lab is virtual, therefore there's no pre-lab today. I hope you enjoy that. That's one less thing to worry about. One less thing for me to grade. I know I enjoy it. And because lab is virtual next week, if we do meet, plan on bringing your computer to lab next week. That's if we meet. Now, granted, without a doubt, I'll be here no matter what. Even if I decide we don't need to meet, I'll be here if you need me. So anyway, any questions about lab, pre-lab this week or lab next week? All right, I've said it before, just a reminder, exam 1, you can meet me online for the correct answers and a 5% boost to that grade. You'll need the correct answers because that is your study guide for the final. Watson is here, right? Watson? Yep. All right, Watson is accounted for. Anyway, so yeah, any questions about exam 1? All right, and actually, again, still, off the top of my head, no one's taking me up on this, I don't think. So since no one has all the correct answers for exam 1, technically I'll still allow it if you retake it. Because the only reason I have to say no retaking it is because once I give out all the answers, then retaking it is simply it's just as simple as plugging in the answers, and that's not figuring it out. So until someone meets me online to get those answers, you can still unofficially go ahead and retake exam 1. Speaking of retaking exams, exam 2 is still open for retaking. And retake it until November 9th, which is when exam 3 is due. So it's the same stuff as usual. Retake it as many times as you want. It's untimed. It's open book. Yeah, you just have to email me when you're done to let me know to adjust your grades. And then exam 3 is going to be due on November 9th. The stuff we're working on now will be due November 9th. So any questions about exams at all? All right, just to remind you, I'm here to help. My office hours are Monday 9 to 10, Wednesday 11 to 12, Friday 9 to 12. I'm here to help you do work. I'm here to help you get feedback. Even if you just need somebody to vent to, even if it has nothing to do with my class, I'm here to help you. So, let me help you. Any questions about office hours? All right. It's almost 8, or excuse me, almost 8:01. So again, well, I'll wait till 8:01 to say it, but are there any questions about anything in general, or do you have your own announcements and reminders? Nothing? Okay. Also a reminder, depending on when I graded your photosynthesis lab, it should be about due. That's 8:01. All right, 8:01, let's get started. So I'm not going to be, that's of all the things, I go ahead and tell you now, that's the one I'm least picky with as far as the due dates are concerned. So I guess what I mean is you should definitely work on getting it done. But, if it's going to be the difference between turning a really crappy lab report in today versus turning a very good lab report in tomorrow, I would choose, the one extra day to work on it. But anyway, any questions about other stuff other than attendance? All right, attendance is normal. It's mandatory. You have to turn it in, and it's graded. Participation's graded. We're going to do what we've been doing recently, which is now when I say the words, they're not voluntary, they're mandatory. So if you're live streaming or in person, you need to write down the words that I say. And the last word, even if I forget to say it, remember, the last thing you need to do is also send me a question about the material covered in this lecture. So yeah. But of course, if you're watching the video at a later date, like I said, when this video started, you also need to send the screenshots. Any questions about that? All right. Also, as a reminder, even though I'm now making it mandatory, you're still getting the extra credit. So as long as you get all the words and you send them in time, you're still getting more than 1 point for participation. Anyway, any questions at all? All right. Well, speaking of attendance, participation, the first word for attendance. Well, I don't want to say it's a word yet. Hear me out. Today is National Food Day. So your first word for attendance is going to tell me either your favorite food to eat at home or the last food you ate at home, either one. Whether you can tell me like, I could be like, oh, we had seafood, or you can get specific and say, we had white chicken chili. I don't care what it is. But your first word for attendance is either your favorite food or the last to eat at home or the last food you ate at home. Probably guess I'm going somewhere with this. Just with the words, nothing else. All right, let's get started. You guys all seem tired. I am too. Hopefully we can wake up. All right, so to remind you, we're done with the first main bullet point of this 3 bullet point chapter. We're done talking about DNA structure and replication. For the most part, for the exam, you just need to know, first of all, the stuff you learned in the previous chapter, chapter 2, when you know the parts of the nucleotides, that the bases and the sugar phosphate backbone, and the fact that A's go with T's and C's go with G's. And you need to know that the enzyme that does DNA replication is called DNA polymerase. So that's the big picture, right? Now we're moving on to this. And like I said on Wednesday, and I'm not going to require this, but my suggestion is to study the big picture and then start adding in the detail. And what I said on Wednesday was to memorize the fact that we go from DNA to RNA to protein, right? And that's going to be one of the test questions. And that's the biggest one. Because if you just happen to remember that it's called transcription when we go from DNA to RNA, or that it's called translation when we go from RNA to protein, but you don't remember that we go from DNA to RNA to protein, that's, I don't want to say useless, but it's not as important. For example, one of the things people, still a few people do, but they say what the COVID-19, the mRNA vaccine is gene therapy because they're injecting mRNA. And as you're going to learn in this section, you're going to learn what mRNA is, and it doesn't work that way. Because what they're suggesting is that we're going from RNA to DNA. That's what gene therapy would be, right? We're injecting RNA, and it's somehow editing your DNA. That's not what mRNA vaccines are, because we don't go from RNA to DNA. There are exceptions to that, and we'll talk about them in this section. But all right, there you go. For the recording, just so I can check back, Smith Still is here. Still has come in. So anyway, let's talk about it. First of all, let's talk about. I don't know if I mentioned this or not. I think I mentioned this, but it's worth repeating anyway. So you already know what a genotype and a phenotype is, right? We learned about that in the heredity, in the genetics chapter. Again, now we're looking at this at the molecular level. So we're looking at it at the molecular level. When we talk about phenotype, another way of describing it is it's the physical traits that are from the actions of a wide variety of proteins. So again, like I keep saying, think about the genetic side. When you talk about a chin dimple or different eye color, or whether how your fingers come together, or whether your little finger's bent or your hitchhiker thumb, all that stuff, that's not necessarily directly a protein thing, right? Or your hair colors, stuff like that. That wasn't on the exam. But the point is, all these different things about you, whether it be a disease or something that you can see, physical traits, they're all due to something being made, right? Your body's making stuff. Your body made who you are, following the directions written in your DNA. And it's enzymes that did it, right? So it's enzymes that are responsible for each one of these little chemical reactions. And remember, enzymes are proteins. So that's how, when we're talking about genes, we're always talking about genes are instructions to build proteins. So don't ever let that confuse you. Don't think to yourself, well, if genes control the directions of proteins or how to make proteins, but we're not all proteins, then how does that work? So now you know, again, I'm reminding you. It's because even if you think about something that's not protein in your body, it's proteins, it's enzymes that directed the manufacture of whatever that thing is. So that's how your DNA, your genes, are in control of who you are physically, right? It's because, again, you have these genes that dictate how to make these proteins, and a lot of those proteins are enzymes, and those enzymes make these very specific chemical reactions happen. But sometimes, as you can see in this one example, and these are just examples, you don't need to memorize them or write them down, but sometimes it actually is protein, right? So sometimes the gene simply says, all right, this is how you build this protein, and that protein helps make up the body of make up the body of your organism, right? So hair is protein, fingernails are protein, muscles, right? So sometimes it is protein, but more often than not, like I said, now I'm just seeing it in writing, enzymes catalyze chemical reactions that are necessary for life, right? So I also wrote a little note for myself, some non-book examples. Melanin is a protein, right? There's a gene that's responsible for that. And somebody actually asked me a question with this, with one of their attendance words. I'm not gonna read all that, but long story short, melanin is the substance that gives skin, hair, and eyes their color, right? So it's the enzymes that are responsible for how much melanin you have. So again, it's genes that are responsible for building these proteins, and those proteins are involved in the manufacturing of the melanin, which affects your color, right? Your skin color, your eye color, your hair color, all that because of genes. So anyway, this is still just kind of an introduction. Are there any questions? Okay, again, like I said, big picture, the first thing you should memorize is that we go from DNA to RNA to protein. That's the most important thing, in my opinion, to memorize. That's the biggest picture. Then once you have that memorized, then let's talk about the names, right? So when we go from DNA to RNA, that's called transcription. And when we go from RNA to protein, that's called translation. As a student, at first, that was a little bit confusing, not confusing to me, but hard to memorize and differentiate when I memorized it. And here's the easy way for me to memorize it. And I'm going to say this again throughout this chapter. Who knows what transcription is outside of biology? Like if I was I don't know, some kind of CEO and I had I don't know, an assistant, and I said, all right, I'm going to write this down. I need you to, or I'm going to say something. I'm going to speak out an email. I need you to transcribe it for me. What does transcribe mean outside of biology? Stover.
It's it's like a person who types out everything that's being said.
Yeah, that would be the person, right? Yeah, exactly. But what about just like, so the action, the verb of transcribing, because you're right, that would be the person, but just the word, the verb transcribe, what does that mean? Like if I'm giving notes and I say, all right, I need, like, if I have one person in charge in this class and I'm giving them extra credit, like, all right, everything I say today, I want you to transcribe it for me so I can post it for the rest of your students. What does that mean when I ask somebody to transcribe something?
Right.
Transfer over, good, we're getting there. In what specific way? I was hoping hopefully you guys are just too shy to say it. Transcribe just means to write something down. So if I'm telling you to transcribe this, that means I say it, you write it down, right? Now, if somebody was in here who couldn't speak any English, and I wish Perez was here. Perez was here, because I know she can speak Spanish. I would say, hey, while I'm speaking, can you translate what I say? And, it's Spanish. That's what going from one language to another, right? So think about that. Hopefully that'll help you memorize this. So for the recording, Jackson is here. Just so you know, what you missed, Jackson, is you have to send in all the attendance words today, and the last word for attendance will be the question. Anyway, so when you're transcribing, think about it. DNA to RNA, that's the same language, right? That's just going from one type of nucleic acid to another. So it makes sense that you would write it down, right? You're just writing it down. You're just going from one, it's still the same language. You're taking something written and writing it on something else. Translation, obviously, is when you go from one language to another. And in this case, we're going from a nucleic acid language of RNA to a protein language. So hopefully that'll help you remember the difference between transcribe and translate. And I'm hoping you guys knew what transcribe meant before I said that. That would help you, but not like in non-biological terms. But anyway, any questions about this? This is still big picture. We're going to talk in great detail about these two processes, and there will be questions about them on the exam. All right. So obviously the relationship between genes and protein is one of information flow, meaning the information is flowing from the code written in your DNA to the code written in the RNA. Finally, that information is translated into proteins, right? This is information flow. So the function of a DNA gene is to dictate the production of a polypeptide, even though we don't get there right away, right? So the DNA gene, again, is transcribed into RNA, and then that information is translated into a polypeptide. And remember when we learned about proteins, as far as we're concerned, in this 100 level class, a polypeptide is a protein, right? So sometimes I'll use those terms interchangeably, but just to remind you the difference. A polypeptide is just a strand of amino acids, right? It's just a string of amino acids. It's not technically a protein until it's folded into its correct shape. And when we talked about that in chapter 2, I used a paper airplane as an example. I said, what is this? It's a piece of paper. And you guys said, that's a piece of paper, and you were correct. And then I folded it into a paper airplane. I said, what is this? And you said paper airplane. So that is correct. But it's also still a piece of paper, right? You guys all agreed. So it's the same thing, right? A polypeptide is just a long string of amino acids. It's not a protein until it gets into its original shape. Even then, when it is a protein, it's still considered a polypeptide, but we also consider it a protein then once it's in the shape. So yeah, this is how this works. This is big picture stuff. If I were studying, if I were you, I would, memorize this slide before you went on and started studying the other stuff. And again, I'm going to mention, or not again, but some of the stuff I'm going to repeat. So I'm going to say it now. I'm going to repeat this again later too. Just so you know why this happens, think about this. Last chapter, I kind of described your chromosomes as books, right? I said you have 23 pairs of books in your genome, right? Inside your nucleus. So you have a pair of each chromosome. Now each one of those books is instructions on how to build a protein. But that's your master copy. So imagine if there was some place on WBSU's campus that had the master copy of how to build stuff, right? That's the only copy. There is no, pretend like there is no internet, all right? So this is your one copy. Now let's say somebody needed a bench built right next to Hamlin Hall. They're going to go into where the books are, the master copies. They're not going to take the master copy. First of all, it's too big. They don't need all those instructions. They only need instructions on how to build that one bench. So you're going to open it up to that page. They're going to write down the instructions they leave, and then they're going to take it out, and they're going to use those instructions to build the bench. That's kind of what happens with your genes. Your genome, your chromosomes, that's the master copy. So you have to keep them safe in the nucleus. You can't take them outside of the nucleus and then follow the instructions on how to build the proteins. And again, your chromosomes have instructions on how to build thousands of proteins. But if you only need to build the one protein, you don't need to take the whole chromosome out of the nucleus, right? Because you only need that one. So again, that's why you write it down into RNA. You transcribe it into RNA, and then that is what leaves and it's translated. And again, I'm going to repeat that. But any questions about this slide? Okay. The next word for attendance is another question. In this case, sort of like the last one, except now it's going to be, what is your favorite food to eat in a restaurant? Or the last food that you ate at a restaurant, if you can't, because if you ask me my favorite, that's almost an impossible question for me to answer. Depends on my mood. And obviously I have no way of checking. So if you're allergic to shellfish and you tell me your favorite food is lobster, it's not like I'm going to say, I called your mom and she said you're allergic to lobster, so that doesn't count. Say whatever you want, but I am curious, so it would be nice to know. Anyway, if there's no questions, let's move forward. So let's talk about how, like, the overview. This was, again, what I love about the old textbook. I've based this off the old textbook because I like the way they present the information better. First, we're going to do this overview of how we go from DNA to RNA to amino acids, right? It's an overview. Then we'll go back and look at it specifically and look at it in more detail. So here we go with the overview. The information that's written in your DNA is transcribed to RNA. And then that information is translated into polypeptides. And then later, and we don't even talk about this process, but later those polypeptides, they are then folded and they assume their final position, at which point they're considered proteins. So again, what we're going to focus on is this idea of going from DNA to RNA, and then that RNA being turned into the information in the RNA being used to build a polypeptide. So any questions about this? All right, here's a very basic picture of what we're talking about. Again, like I said, you've got all of these chromosomes, 23 pairs of chromosomes, safely tucked away in your nucleus where they should be. Those are your master copies, right? So if you have one protein that you need, you're going to go to the chromosome that has those instructions, and you're going to read that part of the chromosome. Because again, you don't need the whole chromosome. You just need that one gene, right? So we're zooming in to this one section of DNA. We read that code. Again, we don't want to take the nuclear or the DNA outside of the nucleus because it's not safe. And it's the master copy, and it's the whole chromosome, so we only need a part of it. We write it down. The same code that's written in the DNA, basically, we write it down in RNA. You'll see what I say. Basically, we do make some changes, but basically, we take that code, we write it in RNA. That RNA goes through some changes that we'll talk about later. It then leaves the chromosome, leaves the nucleus, where there's some other cellular machinery involved that takes that code and reads it. See all these little balls? Of course, that's not what amino acids really look like, but it's a good visualization. Keeps it simple, right? So each one of those little balls is an amino acid. So the code written in this RNA says, all right, bring me this amino acid. Now bring me that amino acid. Now bring me that amino acid. That's all it is. So the code in your DNA, which is then transcribed into the RNA, all it is saying this amino acid is attached to this amino acid, and then bring in that amino acid. That's it. It's just a way of saying bring in this, 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 and this, right? It tells you which amino acids get attached to which. That's all it is. If you download the PowerPoint, you can watch it in action. So like you're going to learn later, this red thing is the RNA. This brown thing is called the ribosome, and you'll learn about that later. This thing here, that's the polynucleotide. Excuse me, that's the polypeptide. So that's where, again, that's what's going to be the protein. And there's a code here, and it'll read the code. That machine will read the code and say, okay, bring me in the next amino acid, and it will. It'll be attached there. And then it'll move. And then it'll say, bring me in the next amino acid, and it'll come in and it'll attach. And that's how it works. It's basically a machine that's adding these little balls to keep it simple. They're not actually balls, because remember, amino acids, all 21 of them, they're different, right? They all have different shapes, different chemistries. So anyway, that's how that works. You can watch it in action if you want. So this is also going to be repetitive. I've already said this. Now I'm saying it again, and now you can see it in writing. What is the language of nucleic acids? All right, we're talking about language because, again, when you're going from transcription is a term that you would use in language, right? Because you're writing something down. Translation, that's obviously used in language because when you're going from one language to another. So it's a great way to look at this. So again, the language of nucleic acids, it's the DNA sequence, right? Those A's, C's, T's, and G's, that's the language. Just like if you were to say, what's the language of English? One of the ways you would describe it is, well, we have an alphabet with A, B, C, D, right, all the way through Z, right? We have all these letters, and that's what makes up our language. We put those letters together in different combinations to make words, and those words mean something. Same thing happens here. We have letters in the DNA, A's, T's, C's, and G's, and we put them together in different ways and different combinations to make different words. And each one of those words says, bring me this amino acid. Okay, now there's a word that says, bring me that amino acid. And there's a word that says, bring me this amino acid. So that's how that works. That's why we say, again, it's a language. And again, this is being transcribed, right? So the language, these letters that are written down in the language of the DNA is transcribed to RNA. The part where it gets a little bit tricky is this bullet point here. And that won't necessarily be a test question, but you'll need to know it because you're going to be asked to do it on the exam. The bases of the RNA are complementary to the DNA strand. So for example, A, C, G, G, A. Right? If that were the DNA code, and then you're going to see this again in writing, but I'm writing it here, that way I can keep it up in the background the whole time I'm talking about this. If that was a DNA strand, and I said, transcribe this DNA into RNA, and you will get a question like that, then you need to define the complementary, right? So you should know by now that A's go with T, but also there is no T in RNA, there's only U. So that would be U. Does anybody remember what goes with C? That one's less tricky. T was tricky because you had to change the T to a U. Does anybody remember what goes with C? And when you think about it, you should, because I just said A goes with T. So there's only one option. If A's and T's go together, what goes with C? There you go, good. G goes with C, G goes with C, A goes with T. Again, there's no T in RNA, so it'll be U. So that's what we mean by they're complementary, right? So the words, these are the words, they're the letters, right? These letters are put together to make words in DNA, and we transcribe it. Of course, the words look different, but they'll have the same meaning, and I'll explain what I mean by that later. But yeah, so that's how this language works. The language consists of 4 letters. For DNA, it's A, T, C, and G. Those are the 4 letters. For RNA, it's A, U, C, and G. So unlike the English language with A through Z, that's a lot of letters. In this language, there's only 4 letters, right? But just like any other language, we put those letters together to make words. And I'm going to come back to that concept here in a second. Any questions about this slide? All right, so that's transcription, right? We're going from here to here. We're just writing down the code, writing down the words. Translation, just like it is in the other terms of this, other senses of the word, it's a conversion, right? Just like if you're not biology and usually talking about translating, usually what that means is you're translating from one language to another. It's a conversion of one language to another. Same thing here. We're converting it from the nucleic acid language to a polypeptide language. So again, these words say, bring me this amino acid. Because remember, polypeptide is just a strand of amino acids, like a chain of amino acids attached one to another in a linear fashion, right? So these words say, dictate which amino acid comes in. 20 amino acids. I think I said 21. I did say 21 earlier. Sorry about that. 20 amino acids. So again, these words dictate which of the 20 amino acids is going to be attached to which. Any questions? All right, and again, this is big overview stuff. We're gonna go into detail on this. Here's a picture from the old textbook. It's good simplification. It's a bit more complicated than the last one you saw, but not that much more complicated. So again, here we have this big long chromosome, this DNA molecule. We've zoomed in on it, zoomed in close enough to where we can see 3 genes. Of course, you couldn't really see them in real life, but it's a good visualization. Now we're going to zoom in particularly to gene 3 to this one small part of it. Right, here's the DNA strand, and there's these letters, right? These letters, AAA, CCG, that's the language, right? Those letters have been put together to make a bunch of words. So we're going to take that information and we're going to transcribe it into the complementary strand of RNA. So A's go with U, C's go with G, G's go with C, right? It just writes it down. And then, and I haven't talked to this word yet, so don't worry about it. I'll officially introduce you to this word later. But each one of these words is 3 letters long, and it says something, right? So this word says, bring me a square amino acid. This word says, bring me a round amino acid. This word says, bring me a triangle amino acid. This word, same as this one, right, says, bring me a square amino acid. So again, this is just a language, just like any other language. It's made up of letters, and those letters come together to make words, and these words tell you which amino acid is next. Also, to remind you, this is a very oversimplified thing. Obviously, this is not like square amino acids, circle amino acids, triangle amino acids. It's just a great way to visualize it. Because if I were to show you actual pictures of what the amino acids look like, unless you're at a higher level of biology or in chemistry, it wouldn't really make any sense to you. It'd just be a bunch of squiggly lines, like, okay, that all looks the same to me. But we're all familiar with the difference between a square and a circle and a triangle and a square and another square. So any questions about that slide? Okay. Nothing to memorize here. There'll definitely be no question where I'm like, remember that slide where I showed you 3 genes? What was the code? Like, I'm not going to ask you to memorize this. Now, granted, there will be a question where I do give you a code like this and ask you to transcribe it, in which case you'll need to be able to do something like that, transcribe it from there to there, but you definitely don't need to memorize that. You just need to memorize, like I've already said, A's go with T, C's go with G. And remember, for RNA, there is no T. There's only a U. If you remember that, and you do need to memorize that, then you can do that. And like I said on Wednesday, just as a hint, because it's not meant to be a trick question, it's actually kind of the opposite. When I give you a question, and I give you a strand of DNA, and I say transcribe it, and that's all I say, you should know that transcribe means to make a strand of RNA. Therefore, when you're looking at this multiple choice exam, anything that has a T in it for that question, you know is incorrect, right? Because there is no T in RNA. So if I'm asking you to transcribe something, there'll never be a T. So that'll save you some time. You can eliminate those questions or those options. Any questions about that slide? Okay, again, well, this is everything I just said, but some of it's just now in writing. So this flow of information that's going from gene to protein, or from DNA to RNA to protein, right? This is based on a triplet code, which is another way of saying in this language where there's 4 letters, those 4 letters are brought together to make words. In this language is different than the English language in that all the words in this language are 3 letters long, right? They're all 3 letters long. Unlike, think about, the English language. You've got a word like A versus a word like supercalifragilisticexpialidocious, right? So that we have all kinds of variations with the size of our English words. And in this particular language, they're all based on 3 letters. All words have 3 letters, and you do need to know that. So the instructions for the amino acid sequence are written in the DNA and the RNA as a series of words, and those words are called codons. So again, it's everything I said on the previous slide, but now you're just seeing it in writing. So we have words for these words. We don't say, when we're talking about this process, we don't say, hey, what word tells me to bring in this amino acid? And what word tells me to bring in that amino acid? We would say, which codon tells me to bring in this amino acid? Which codon tells me to bring in that amino acid? So the codons in the DNA, like this up here, are transcribed into complementary codons in the RNA, like this right here, right? So again, like I said, these codons are the words that are 3 letters long. They're always 3 letters long. So there's a word, right? There's 2 words. I wrote 2 words in that example. Yes, so those words are transcribed into those RNA words. And then on the picture here is this code right here will be translated into amino acids, right? This one says, bring me a particular amino acid. That one says, bring me a particular amino acid. And later in this chapter, I'm going to teach you how to do that too. So yes, one question is going to say, here's a strand of DNA, transcribe it for me. In which case, this would be the answer, right? Because you're going, when I say transcribe, you're going from DNA to RNA. There'll be another one when I say, here's a strand of DNA, translate it for me. That was admittedly a little bit harder because at the very least there's more steps. Because to translate that strand of DNA, first you have to transcribe it to RNA, and then once you figure out the RNA code, then you have to decode it to figure out which amino acids. And we'll get to that later, maybe today, possibly Monday. All right. Any questions about that? All right, again, same picture that we saw previously. There's code in the genes, right? The codes are these letters written in the DNA. And each one of these words, aka codon, is 3 letters long, right? Each one of those words dictates a particular amino acid. All right. So speaking of which, let's talk about the genetic code a little bit. So we're kind of taking a step back. We are going to go back to this bullet point, right? We are going to look into more detail about how we get from nucleotides to amino acids. But before we do, let's talk about the genetic code. And this to me is where it gets a little bit interesting, I think. The genetic code is a set of rules that converts RNA sequence to amino acid sequence. So that's what the genetic code is. So again, when I said transcription is when we go from DNA to RNA, translation is when we go from RNA to protein. When I said that, right, that translation, it's decoding it, right? Because these are codons, right? We call these words codons. So the genetic code is basically the decoder. It's how do we figure out what UUG means? How do we figure out what CCU means? Now, if you're good at math, I could have asked you, knowing that there are only 4 letters in this language, and knowing that the words are only 3 letters long, how many words are there in this language? And you could have figured it out if you did the math. But here's the answer, 64. So this language has 64 words. I'm probably not going to ask you that. That's what that little star was for, is to remind you that I'm probably not going to ask you to know that number. But just in case, study that if you feel like that when you're studying, if you feel like you memorized everything else, maybe add that one on at the end. Yes, there are 64 triplets, 64 codons, 64 words, right? I said the same thing in different ways. Of those 64 words, 61 of them code for a specific amino acid. So 64 of those words, or 61 of those words says, bring me this amino acid, bring me that amino acid, bring me this amino acid, right? 61 of those do that. 3 of them are just called stop codons. So that's the code that says, okay, stop. We're done with this. We're done making this polypeptide. The job is done. Those numbers, I'm definitely not going to ask you to memorize 61 and 3. But if this wasn't an 8 a.m. class, and if you guys weren't taking naps or looking at your phone, hopefully you'd be a thinker and you'd say, hold on, you're saying there's 61 words to say bring in this amino acid or that amino acid. But earlier you just said there's only 20 amino acids. So how is that possible, right? How do we have 61 words that say bring in this specific amino acid, but there's only 20 amino acids? And this is how it works. That looks confusing at first, but it's really not. The thing is, each amino acid, each of the 20 amino acids, there's more than one word that says bring in this amino acid. Well, for some, yeah. So for example, alanine, right? Well, let me back up and start a different way. Then I'll come back to that concept. So like I said, there's going to be a test question, at least one, where I give you a strand of DNA, and I'm going to say translate. And what you're going to do is you're going to know, using these base pairs, right? A's go with T's, C's go with G. You take that DNA strand, you'll transcribe it into RNA, and then you can use that, and I'll provide you with this wheel. And it looks complicated, but it's not. Let me show you. You'll use that to decode it, right? To figure out what amino acid. So UUG. It's very simple. You start here and work your way out. All right. UUG. Leucine. Abbreviated LEU. So that would be LEU. Then we have CCU. So again, start here in the middle. CCU. Proline. Abbreviated PRO. And that's how that test question is going to go. So again, you don't need to memorize this wheel, but you need to know how to use it. You're going to have a 3-letter code, and you're going to start in the middle and work your way out, right? GCC, alanine. AGC, serine. That's how that's going to work. It's going to be that simple. But anyway, going back to my other point, there's only 20 amino acids, but there's 61 words that code for amino acids. Now, generally speaking, there's more than one word, right? So again, using alanine as an example, the words that say bring in an alanine are GCG, GCA, GCC, and GCU, right? Any of those words will say bring me in alanine. Of course, not always like that. Like this one right here. And that's always the start codon, but you don't necessarily need to know that either. But anyway, that's the point. Generally speaking, that's how we have 61 words in this language that code for a specific amino acid, but only 20 amino acids. That's how it works. So are there any questions about this? I know it looks complicated, but it's really not when you think about it. Just start in the middle, work your way out. All right. There's no questions. We'll move forward. The next question for attendance, again, this will be another question since it's National Food Day. And since we are 1 week away from Halloween, what is your favorite candy? Or the last candy you ate, either one. All right, moving forward. Now, this to me is the more interesting part. The genetic code is universal. I'll put a little star next to that because there are very few exceptions. So you can look that up for extra credit if you want. Don't forget to include your sources, a link to your source where you tell me about what the exception is to what the genetic code is universal. But as far as we're concerned, it is genetic. It is universal. Right? Whether it's a bacteria or a human or anything in between, it is universal. And this is basically where GMOs come from. And I know some people are scared of GMOs, but they're not always scary. I can see how some of them can be scary, but some of them are just really benign, like completely not even scary. I don't even see how people are scared of it. For example, who here has ever heard of super rice? They don't sell it much here. But anyway, I'm sure you know that rice is a staple food for a lot of people, especially in like Asian and African countries. So that's where they're getting most of the nutrients. But rice does not produce vitamin A. Vitamin A is an important vitamin. So what they did, since the genetic code is universal, they took that code, the genes that are responsible for producing vitamin A, which was found in carrots, they took it out of the carrot genome and they put it into the rice genome. And now this rice, called super rice, produces vitamin A. That's one example. Because the code is universal, we can do that. How many of you guys know people who are diabetic and have to take insulin? A couple of you, a few of you. That is because of this. We took the human code that says this is how you produce insulin, and we put it into bacteria because bacteria are easy to grow, and bacteria follows it. They don't know that that's a human code because the code is universal. So all these bacteria are producing this insulin, which is then taken out and purified, and that's what the humans get it from. It used to be taken from pig spleens. So this is obviously a lot better. Anyway, those are just examples. You don't need to know any of those examples. Here's some pictures I found that were not in your textbook. It might be a little bit hard for you to see. Yeah, it's almost impossible. Almost it's just like there's something wrong with the projector and the fact that we have a light coming in. But these cows, man, won't that be embarrassing? These pigs are glowing, right? That's a glowing snout, glowing hooves. And I don't remember why they did it scientifically. I'm sure they didn't just do it just to do it because that wouldn't be ethical. But anyway, they're glowing because somebody took genes from jellyfish, glowing jellyfish. They took the genes responsible for those jellyfish glowing, and they inserted them into the genome of that one pig. Yeah, just that one in the middle, and it is now glowing. Same thing with that rat below it. That's a little baby rat or mouse, I forgot which. Anyway, it's also glowing because they took the gene from those jellyfish that make the jellyfish glow, put it into these mammals, and it made those mammals glow. In theory, you could do that to a human. Ethically, that would be, I doubt anybody would do it because it would be wrong, but you could do it. Speaking of what you could do, that's what this picture is. This picture is hypothetical. I don't think anyone's ever done this yet, but I've heard people talking about it. The technology's there. We can easily do it, but there's other ecological implications. But anyway, what this is a picture of is glowing trees. Someone had the idea, or has the idea, if we take those genes that make, the jellyfish glow, or maybe even a lightning bug, whatever, and insert it into the plants, then the plants will glow. And that is very possible. So if you're into that, you could write papers for extra credit about that. Look up real life examples, and this is one where you wouldn't even need sources. You could just use your imagination. It's like one time you could just write in biology class about just something you imagine. Like, for example, we could do this, and I think it would be a money-making venture, but I've already said it on video, so if somebody takes my idea, it's too late. You could take the genes responsible for making THC, which is the stuff in marijuana that gets you high, right? Take those genes and put it into everyday food items like tomatoes or onions and peppers. So then, for example, like if you live in a state like Washington where it's completely legal, you can make some salsa that'll get you high, right? There's a good market for that. But anyway, yeah, you can write a paper if you want to and just get creative. Like, what if we took a gene for this and put it into that? So have some fun with it if you want. But anyway, that's just some pictures, some examples. I'm not going to ask any questions about that. Point is, again, the genetic code, again, it's built with 4 letters in the language. Each word in that language is only 3 letters long, and there's only 64 words, 61 of which code for a particular amino acid. 3 of them say stop. And again, it's the code, those amino acid codes that get translated using that wheel that I showed you to dictate which specific amino acid goes where. That being said, now we're done with the overview. Now we can finally dig into the details. Now we can really talk about how we go from DNA to RNA, which you already know is called transcription. So let's dig into transcription and talk about how that works. That might be the last thing to talk about. So we have 2 minutes left. So anyway, transcription, again, memorize the big picture before you start adding the details. So if you don't know that transcription is going from DNA to RNA, then the rest of the stuff I'm about to teach you really doesn't matter. You need to know if transcription is going from DNA to RNA. Right? So make sure you know that. Obviously, it's the DNA where this happens, right? The DNA is the code. So this is where the whole thing's going to start. More specifically, the DNA strands opened up. And this should sound familiar, because that's exactly how it happened when we talked about DNA replication, right? When we talked about what happens during S phase of interphase, and that DNA polymerase comes in and reads the code to make another new strand of DNA. It's exactly what's happening here, except now we're not making DNA, we're making RNA. But still, you have to open up that strand of DNA so you can read the code, right? So you can read those A's and T's and C's and G's. You have to open up to read it. Once it's separated, then we have an enzyme come in and read the code. One of them, and I wish there was a picture. Even that picture sucks, so I'm not gonna wait on that one. So if you had a strand of DNA, A, C, C, G, T, A, C, whatever. I'm not even gonna draw this because I don't want to waste your time. But if you're gonna open it up, right? So obviously, yeah, this is DNA. You're gonna have a T here, right? G, G, C, A, T, G. Anyway, that's a really horrible picture. I'm not an artist. But what you've done, like I tried to describe here, is you've opened up the DNA strand, exposing the code. And what all this is saying is like one of these codes is going to be used. I'm just going to hypothetically say it's this one. This one's going to be used, and this one's not. For the exam, that's not important at all. I'm not going to have any test questions where you need to know that. That's just for your own knowledge. What we're saying here is when you open up the DNA strand, obviously, you're going to have two codes, one on one strand, one on the other. All we're saying here is that only one of them is used, the other one is just completely ignored. So any questions so far? All right. So the way this happens, and we're going to go into more detail. This one bullet point, it's more like an introduction to other stuff that you can learn. What's happening here is that, like, if I was the machinery doing this, I would bring in, like, all right, that's a C, right? So I need a G. So I bring in a G, and boop, it would get a hydrogen bonded to it. At which point, I would then covalently bond it to the other one that was already there. And then next, I'd bring in, all right, there's a C, I'd bring in another G, and it would hydrogen bond to that, and then covalently bond to the strand that was already there. That also is something I'm not going to go into detail on the exam, but I just want to let you know how it works. So as far as the exam is concerned, there's a lot of extra information on there that you don't need to know, but that's okay. Obviously, I have a pin here, hopefully to remind you that we're transcribing something, right? We're writing something down. That's all the pin is for. Any questions so far? Okay, again, like I've already said, the RNA nucleotides follow a base pairing rules, right? So again, A's go with T's, C's go with G. The only tricky part is you need to remember RNA does not have a T, it has a U. So again, if I ask you to transcribe a strand of DNA that has an A on it, right? If it was another strand of DNA, T would go with A, right? T's go with A. But again, RNAs don't have T, they have a U instead. So boom, we have a U instead of a T. All right, now the second enzyme I told you about earlier on Wednesday. I said there's going to be two enzymes you need to know in this exam or on this chapter. The first one we've already talked about. It's the thing that makes the new DNA, and that's called DNA polymerase. The other one that makes the RNA is called RNA polymerase. Someone's going to miss that question, and it's going to make me sad because it's in the name, and they're still going to confuse the two. There'll be a question where it says, "Hey, you know, which enzyme is used in S phase of interphase to make a new strand of RNA or DNA?" And someone's going to say RNA polymerase. I'll shed a little tear because it's in the name, right? DNA polymerase is the thing that makes the DNA. RNA polymerase is the thing that makes the RNA. Know that for the exam. Also, I want to point out, just like I did with the DNA polymerase, this is a 100 level class. We are really oversimplifying it. This picture is not from your textbook. It's not from the old textbook either. But if you look closely, what we're looking at here is RNA polymerase. It's attached to a strand of DNA, right? That's what this is. This long thing is the strand of DNA. This big blob here is all RNA polymerase. It's attached to the DNA. And this squiggly thing coming off is the new strand of RNA that it's making. But if you look closely, you can see this RNA polymerase is actually different colors, right? That part's green, that part's blue, that part's also blue, that part's purple, right? They're different colors for a reason, because in all actuality, when you're not taking 100 level class, you would realize that RNA polymerase is actually a team of proteins working together, right? So we are oversimplifying things. And I say this because if you want to look things up, hopefully you will, and you'll say, oh, wait a minute. He only said RNA polymerase. What are all these other words that I'm reading? So I guess the point is, just like with the last thing with the DNA polymerase, if you look this up elsewhere, and you see other words that you don't recognize, you can just ignore them as far as the exam is concerned. Of course, excuse me, if you want to learn about it, you can ask me and say, hey, I was watching this little video that shows how this works, and it kept bringing up helicase. What is helicase? And I can explain it for you. But again, for this 100 level class, we're just oversimplifying. We're calling this whole machinery RNA polymerase. Any questions? All right. This picture is also an oversimplification. It's more of a drawing of what you just saw, right? Here's the long strand of DNA. Here's the RNA polymerase that's attached to it. Part of the team of RNA polymerase has opened up this strand of DNA so we could read the code on one strand and ignore the other, right? So it's reading the code. It's reading there's a T. So like I said in the previous slide, that A is going to hydrogen bond to the T. And hopefully you can visualize. Imagine that just fitting in there. And once that A is hydrogen bonded to the T and it's all fitted in there, Then we can make that official connection between this A and that A, right? We could covalently bond that. Then this U is going to come in and hydrogen bond to that A. And once it's all nice and flush next to that, this A, we'll do the covalent bond that brings them together. That's how it works, right? We open up the DNA, RNA polymerase brings in these codes one at a time, and that's how it works. And again, to remind you, even though I just said this, you don't need to know it for the exam. Yes, technically we have a hydrogen bond first and then covalent bond, but that doesn't matter. The point is, RNA polymerase is opening up the DNA to read the code, and it's bringing in the complementary nucleotides one at a time as it's reading the code. And then obviously, as you can see here, something eventually closes it back together. Some part of RNA polymerase, then after it's opened it up to read the code, closes it back up. So any questions about that picture? All right. Here's where it gets a little bit more tricky, right? So we know that transcription is going from DNA to RNA. Now, to make life worse for biology students, the scientists have sub-categorized that. So the process of transcription has three parts. There's initiation of transcription, RNA elongation, and termination of transcription. What I do like about this, in my opinion, is they're all it's in the name, right? Initiation of transcription, we're starting transcription. RNA elongation, we're making the RNA strand longer. Termination of transcription, we're ending the whole process, right? It's nice and simple. So memorize that over the weekend. And then on Monday, when we come back, we'll talk about what each one of them does, and I give you some more words you need to know. The last word for attendance, again, don't forget, send me a question about the information that was presented in this lecture. That's it. You guys have a good weekend. I'll be online for office hours if you need me.